Hi, everybody. I'm Mary Beth Horsington, and I'd like to welcome you on behalf of the Strathmore Speaker Series. Thanks so much for joining us here tonight for another informative program, the 9th event of our 2022. It's not the 2022 season, but for the year 2022. Our partner on this endeavor is Onondaga Free Library. They provide the technical assistance we need to bring you this program, so we thank them for their expertise and generosity. The Strathmore Speaker Series is made possible through the support of the City of Syracuse, the Gifford Foundation, the Greater Strathmore Neighborhood Association, and donations from people like you. If you would like to support this effort, go to our website, strathmorespeakers.com, and you'll see a donation button on the landing page. Every dollar is greatly appreciated. We are looking for new members to join the board of the Strathmore Speaker Series, so if you'd like to join us, let us know. You don't have to be a Strathmore resident. We welcome anyone who might want to share their talent in bringing this informative series to our community. Our guest speaker tonight is Dakota Matthews, Molecular Lab Manager at SUNY ESF's American Chestnut Research and Restoration Project. His topic will be a forest reborn, reviving the American chestnut. He'll talk about the history of this remarkable American chestnut, which fell victim to a blight in the early 1900s. We'll learn about the pioneering work this project is doing to achieve the goal of growing 10,000 blight-resistant American chestnut trees over the next 5 years. A brief question and answer session will follow his presentation. And now, please welcome Dakota Matthews.
Thank you, Mary Beth, and thank you, Scott. Thanks for having me, everybody, and I'm excited to talk about our project at SUNY ESF. Today I'm going to be talking about the American Chestnut Research and Restoration Project at SUNY ESF and our partners, the American Chestnut Foundation, as well. The American chestnut was once the predominant eastern hardwood deciduous forest tree. Of all the hardwoods in the eastern U.S. forests, it accounted 1 out of 4 trees in its dominant range. And here you can look at a picture of Shenandoah National Park in 1912. This is our classic sharing picture to demonstrate the predominance of this species. They would call it snow in July. You can see all the white tipped trees. Those are American chestnuts. Their flowering male catkins are these yellow whitish branches that produce American chestnut pollen, and they distribute it widely throughout the range. So when you would look at a chestnut forest, you would see what we would call snow in July. It has a variety of forms and traits. To the left, you can see an old picture that we also love to use because it just demonstrates the timber type American chestnut of an old growth forest. They were called the Eastern Redwoods. They got hundreds of feet tall, huge diameter, very straight grain growth. But there's also a variety of American chestnuts that are very much street and orchard tree. The spreading branching pattern is also in quite a few old poems from the early 1800s, 1900s. It was a very, very valuable tree. The wood products were used for fence posts, barns, and cabinetry, furniture making. It was very easy to work with, very rot resistant, and you didn't have to treat it with any chemicals or byproducts to have a long-standing structure. It is highly sought after in resale markets and antiquing for woodworking, and you could sell a decrepit barn made of chestnut wood for a good penny, still to this day. Agriculturally, it was highly valuable as well. For hog farms specifically in the Appalachians would let their hogs go into fenced American chestnut stands in the fall to fatten them up for winter, free crop. And it is also a great gluten-free food source for humans as well. We talk about Dr. Powell who is the leader of our project. He has celiac, so we often are jokingly saying that we're restoring this just so he can eat some bread products that aren't filled with gluten. And you can make a chestnut beer. You can make a ton of different chestnut things as a substitute for gluten-containing foods. There are even some old medicinal uses, chestnut leaves being steeped in teas for a variety of ailments back in the day. That has not been tested, but it actually makes a very good chestnut tea just by itself for tasting. And then there's also just a bunch of social and historical aspects to the value of the American chestnut, whether it's the chestnuts roasting on an open fire. Any city on the East Coast has a Chestnut Street, just like you would see in Oak Street. It was a huge part of our early American culture. It was also a huge part of Eastern US forests as a keystone species. It provided what we like to call environmental benefits from bees to bears. So even the smallest organisms to the largest. Like we said, they're highly nutritious and would provide a stable mast crop each year, unlike oaks, where their mast crops can vary depending on if it's a masting year or not. The leaves would provide quite a bit of nutrients for forest ecosystems, whether they be vernal pools or lining the creeks of forest streams for brook trout, things like that. And it is believed that 5 species of insects are believed to be extinct as a result of the chestnut's demise. You can look them up on the IUCN Red List. So what happened to the American chestnut? Chestnut blight. It is a fungus that invades a wound on the stem of the tree. It colonizes that wound and begins producing what we call a virulence agent, oxalic acid. The oxalic acid kills the living tissue of the American chestnut, and the fungus proceeds out from the wound site, digesting and eating the dead tissue. So it is a necrotroph. It feeds off of the dead tissue. And this caused quite a big panic across the East Coast. There were tons of news articles and stories throughout the early 1900s of the chestnuts being doomed and face destruction. Here is a look at a chestnut stand after chestnut blight moved through. As you can see, the fungus has killed all of these trees. And what it does is the fungus making that canker will girdle the tree all the way around and basically starve the upper top area of nutrients, and therefore it dies back and not survive. The chestnut range went all the way up into Canada and Maine at the uppermost regions of its historical range, and all the way down to Florida, Mississippi, Arkansas. But the predominant range was New York down through Tennessee, the Appalachian area. The Japanese chestnut began to be imported in the late 1800s as an ornamental tree. You can see the Japanese are a great orchard tree for nut production. They're also quite beautiful. They were imported, and we believe they were first imported through the New York ports, which is where the chestnut blight was first discovered at the Bronx Zoo. And throughout 50 years, the blight fungus transferred throughout the natural range of the American chestnut, killing both the American chestnut and its relatives in the chestnut family, Allegheny chinkapins and Ozark chinkapins, which are a smaller understory forest tree, and killed over 4 billion trees in less than 50 years, which was devastating. So chestnuts are lucky, and the reason they're still around today is that they survive by the root collar. So once that upper portion of the tree is starved off and girdled, the tree can still survive by popping up new sprouts from the root collar. So that's how we still have chestnuts today. The issue is that they never reach sexual maturity. So once a wound is colonized by the fungus, that tree is basically gone and it needs to send up new shoots. So all of the trees that we have today that are still surviving across the East Coast are root systems that are over hundreds of years old, but they never reach sexual maturity. So the upper portion of the tree doesn't last longer than 7 years, 9 years at the most usually. But there are some isolated trees. So you can see in this picture here from an aerial shot that there's one lone chestnut in this stand. And those trees are just lucky. Whether the blight is not around or it's relatively undamaged, because the fungus can't invade the stem of the tree without a wound. Historically, there were quite a few attempts to try to rescue the tree. In 1904, this is an image of one of their trees at the Bronx Zoo, where they are just painting and spraying fungicides all over the tree. It's kind of hard to see, but as you can see, there's no personal protective equipment, so those people were just poisoning themselves for no reason, which is a shame. The fungicides have a hard time penetrating the tree because the fungus grows underneath the bark, so it's not very effective to treat the chestnut blight with fungicides, which they found out. They also did a classic technique, which they're also trying to do with the ash tree right now with emerald ash borer, where they basically clear out chestnuts for or ash trees over a swath of 50 or so miles, where they create this barrier, and they attempt to preserve trees in a different part of the range by not allowing the spread of that fungus by removing the host. Unfortunately, the American chestnut, or the Cryphonectria parasitica, the blight fungus, lives on alternate hosts as well. So it survives on oaks, it creates small cankers, but it doesn't kill the tree. So no matter how much removal and barriers that we try to erect, unless we were to just clear cut everything, it wouldn't have worked. And they didn't know this at the time as well. Then we also attempted to replace the American chestnut with a timber type Chinese chestnut. So reintroduce a chestnut species that previously were only being used for ornamentals or orchard type trees. Unfortunately, they cannot compete for the overstory in eastern US forests. So despite the many plantings and the widespread effort, the trees did not survive long. And it wasn't necessarily because of the blight, they just couldn't achieve the canopy to actually dominate in an eastern US forest. This is the most creative attempt to save the American chestnut, and I love to share it because they basically just tried to turn these trees into hulks. They blasted trees with gamma radiation and thermal neutrons, which I don't know what that means, and basically trying to cause random mutations within the genome that would confer resistance to the chestnut blight. And as you can see, the more gamma and more thermal neutrons that they added, the germination rates just went down. Some of these trees are still alive in some plots, but they're not necessarily resistant. They're just well taken care of. Unfortunately, that didn't work as well. And then biocontrol. This is probably the most promising aspect when it comes to demonstrating actual way to manage the blight in forested systems. Hypovirulence is basically the introduction of a virus to the chestnut blight, making it less virulent, making it less able to kill its host species. This works really well in Europe because there's a less diverse group of Cryphonectria strains throughout their natural range. And that, in combination with the somewhat resistance that European chestnuts have, they've been having pretty good success with introducing hypovirulence to blighted trees and letting them survive. Unfortunately for us, because we imported so many different chestnuts from so many different ranges from Asia, we have a very, very diverse group of Cryphonectria strains throughout the eastern forest, and hypovirulence is strain specific. So it's not a functional way to restore the American chestnut in eastern forests when it comes to restoration as a wild type tree. There's other things that you can do to maintain American chestnuts to protect individuals. One of the things that we find to be pretty unique is a soil compress. So we're basically creating a mud pack where we wrap that around a canker, and the microbes in soils actually outcompete the Cryphonectria parasitica, and it can actually kill off the blight and save that tree or save that canker. Unfortunately, you can't just wrap American chestnut trees from top to bottom in soil to save them. It's not a viable strategy. And just because you heal one canker doesn't mean that there's fungus present somewhere else on the tree. So here's a picture of a soil compress. You leave it on for one season and it usually heals up the canker quite nicely. So after a century of unsuccessful attempts to contain and preserve American chestnuts, what are some options that we have? Well, one of them is chestnut hybrids. So hybridizing with Europeans, Japanese, Chinese chestnuts. This is a viable strategy for ornamentals crops. There are great traits in all of these trees that provide a great strategy for, you know, if you want to plant a tree in your backyard or you want to plant a street tree that's semi-resistant to the chestnut blight. However, it's not, as we demonstrated in these previous slides, a viable option for restoration, because our goal here is to replace this keystone species or reintroduce it to its natural range and provide that economic and ecosystem value that it once provided to U.S. forests. And part of the reason it's not suitable for restoration is that these strains of American, or these varieties of chestnut are greatly diverse and have evolved over millions and millions of years in completely separate ecosystems and environments. And that's how the Chinese chestnut is resistant. It co-evolved with the Asian chestnut blight. So it is not a viable strategy when it comes to trying to restore a natural tree to eastern US forests because they are so diverse. And there are also some downsides with hybridization in plants in general, and we have seen this in American chestnut as well. Hybridizing two different species that are sexually compatible causes huge changes in the genome of the original species you're trying to restore. Genome shock is a big thing where some trees just don't perform. There are huge rearrangements, transpositions, and genome size changes. So you're essentially creating a completely new tree when it comes to how big is their genome. And it also can increase the mutation rate in certain varieties depending on the plants that you are crossing. And this is just a quick visualization of, you know, how different some of these trees are. On the left, we have Castanea dentata, which is American chestnut. We have sativa, which is the European chestnut. And then below, we have some understory trees, the Pumila ozarkensis, Seguinii, and Henryi. Those are all different chinkapin species, but they are in the chestnut family. And then we have Millissima and Crenata, which are Chinese and Japanese chestnuts. So there are several documented risks that throughout the hybridization process that we have found in American chestnut crosses with Chinese, Japanese, Europeans. Reduced growth and intermediate traits. We often lose quite a bit of the American traits that make it competitive in eastern US forests, whether it be the ability to reach the canopy, shade tolerance, and nut production. There's documented male sterility between American and Chinese chestnuts when crossing them. So that's an issue when you're actually trying to repopulate a species. Cracked bark between American and Japanese, that's a difficulty as well because we do not want any compromises in the bark for the fungus to actually enter the tree. Internal kernel breakdown is basically the inside of the nut rots away between those crosses, so its offspring are not viable. And many of these can be managed and prevented and bred out in subsequent generations, but they will always come back at some point if the right crosses are made. So if we're trying to repopulate a eastern forest with naturally reproducing species, it's going to be very difficult to manage those in a wild setting. So hybrids are not super suitable for restoration, but there's nothing wrong with them when it comes to planting for yourself or orchard type species, and which is why we went a different direction for our restoration. One of the ways, and this is being done at the American Chestnut Foundation, is through a backcross breeding program. The backcross breeding program is basically crossing a resistant Chinese chestnut with a desirable timber type American chestnut. And so what we get is an F1 cross. And basically that's half American and half Chinese. And then we outcross to American trees throughout, and we breed out unwanted traits. So we select for resistance, we select for nut production, we select for that straight, fast American growth. And we keep crossing with Americans back, back, and back. So that's why it's called a back cross. And we eventually get to a B3, F3 generation, which is 15/16 American chestnut. And hypothetically, with all of these controlled crosses, we are selecting for disease resistance at the highest level. So we're hoping in that 1/16 Chinese set of genes that's in that backcross program, that those are the genes that contain resistance to the chestnut blight. And this was a great idea, and people put quite a bit of work into this. Unfortunately, Jared Westbrook, who is the head scientist at the American Chestnut Foundation National Branch, has recently published a paper discussing that we've actually kind of figured out, due to both these crosses and genomic sequencing, that inheritance is polygenic. So that basically means that since the Chinese chestnut co-evolved with the chestnut blight over millions of years, it developed multiple different genes that all work in tandem together to provide resistance to the chestnut blight. So it's not just a single trait. So the likelihood of us being able to get all of those traits into just that 1/16 Chinese for restoration is very difficult, and we haven't been able to do it in the years that this program has been going on. And we've found that the best intermediate resistant trees that are also the most American that we can generate at this time is around 70%. So we're not actively moving forward with this when it comes to the sole resistance, but they have been able to develop resistance to other chestnut diseases through this program. So they're working closely with us, and we're working closely with them to cross some of our transgenics with their B3, F3s. So we decided to move on to genetic engineering. We did this for several reasons, but I want to give an analogy here to explain kind of the purposes of and the rationale of choosing this method. So chestnut has approximately 30,000 gene pairs, and hybrids have half Chinese, half American genes. And backcross breeding program, like I said, has produced B3, F3s that are 1/16 Chinese. So if we compare that to a book, a backcross tree has approximately, with a book that has 30,000 words, approximately 10 pages or 1,800 words would be in Chinese. So it would be very difficult to glean the plot of the book, the ending, maybe the beginning of the story, the background, if 10 pages or 1,800 words were in Chinese. Now, also, each time we do this, it's random. We don't know where those Chinese words are going into this book. So it's a very difficult thing to actually cross a backcross tree that contains all of the genes you want, but still retain all of that story or all of the American chestnut traits that we want. So we want to insert the genes that make me more resistant or the words in the book, but we might also get traits that we don't want. Make me shorter, larger nuts, slower growing, etc. So that's the analogy for the Backcross program. For us, what we did was this same book that has 30,000 words. Instead, we used genetic engineering through agrobacterium-mediated transformation to make a very small change to this book. So we are only adding 2 words, the resistance gene and the driver of that gene. So of Henry David Thoreau's Walden Life in the Woods book, which is approximately the size of the American chestnut genome if you compare genes to words, we're, instead of changing 10 pages and 1,800 words randomly, we are inserting two genes. And instead of it reading, "It was very exciting at that season to roam the then boundless chestnut woods of Lincoln," it is now changed to the blight-tolerant chestnut woods of Lincoln. So we are retaining 100% of the American genes, and we're just adding an extra 2 to confer blight resistance. So now that we have the methodology down, how do we identify what genes would be best to use, or what gene would be best to use to confer resistance? So we went about this using a mechanistic approach. So like I described earlier on in the talk, the chestnut blight invades a wound on the tree and starts secreting a virulence agent called oxalic acid. Oxalic acid has a pH of basically battery acid, and it kills the tree tissue. So we thought to ourselves, let's try to find a gene that can somehow disable that function or reduce the amount of acid. Because if the tree can't kill, or if the fungus can't kill the tree, the fungus will just survive on the canker, sort of like it does on chestnut oaks, or not chestnut oaks, oaks or Chinese chestnut. And so what Dr. Powell found was an oxalate oxidase gene from wheat. It's a ubiquitous enzyme in many plants and fungi, and it has quite a history in quite a few different species. This is something you eat every day, most likely at breakfast, in any type of cereal, bread. It's in strawberries, bananas, stuff like that. And what oxalate oxidase does is it's generated within the cell, and then it's exported outside of the cell and glycosylated or attached to the cell wall of the plant. So it's outside of the cell. And what oxalate oxidase does is it detoxifies the oxalic acid and turns it into hydrogen peroxide and carbon dioxide, both of which are beneficial to the plant. The hydrogen peroxide is actually a cell signaling molecule in all plants, and it's actually responsible for quite a few disease and wound responses, excuse me. So we're actually providing a beneficial byproduct to this plant by producing hydrogen peroxide by detoxifying oxalic acid. It is not a pesticide. We want to make that clear. We're not doing anything to the fungus. We're not killing it. We are not altering it. The only thing that we're doing is using this enzyme to not allow the fungus to kill the tree. And essentially it goes from a necrotroph, which kills the tree, to a saprophyte, which is just a disease that lives on the tree, but they coexist. And the huge aspect of this that we are very excited about is that because it doesn't kill the fungus or alter the fungus, there's no or very little selective pressure for the Cryphonectria parasitica, the blight fungus, to evolve a more stronger response to eventually kill resistant trees. We are also looking at several other genes now that we've demonstrated resistance with our American chestnut transgenics with OXO, to kind of stack and make our resistance even greater. So we're looking at lactase genes, which are responsible for helping form lignin around the wound site of the canker. So that basically just forms a barrier around the fungus, kind of isolating it a little bit more. And PGIPs, which are a gene I will not bore you with. And like I said, it's ubiquitous. So these are some of the common names, or some of the common species of plants that you would eat where oxalate oxidase is present and naturally there. Like none of these are transformed like our American chestnut tree. So like I said, you probably eat it every day. And then these are natural or wild and ornamental plants that coexist or used to coexist with American chestnut in the natural range. So we're not introducing a novel enzyme to eastern US forests. It's in a ton of grasses. It's in fungi and bacteria. And it's also, we believe it originally evolved from mosses, which were the first plant-like organisms to colonize the world. So how do we get the oxalate oxidase gene into American chestnuts? Well, the first thing we have to do is isolate immature American chestnut embryos. So what we do is we collect immature nuts from our American chestnut trees, harvest them, peel them, and try to extract the embryos from the nuts. So the embryos are up here in the upper part, and if you get them at the right time, you can continuously multiply those embryos in artificial media that basically makes them think they're still in the nut, and they will constantly reproduce. And that is what generates an American chestnut tree. And this is a wildly difficult process. It's about 1 in 1,000 that are actually successful, and it also depends on the variety. So initially when this project started, it was just, can we actually even get to that point? And it took a very long time to actually start producing embryos that we could use. So this is what a somatic embryo looks like, and we keep multiplying that and multiplying it and multiplying it, and then we put it through an agrobacterium mediated transformation. So agrobacterium is a natural genetic engineer. It forms crown galls on the trees in its natural state. So if you're walking out in a forest and you see a tree with a giant, what looks like a giant wart on the side, that is formed by agrobacterium. And what they do is they insert genes into plant cells to have the plant cells produce a food that only they can digest, that's not usable for the plant. So what was discovered in the 80s was you can actually make a plant transformation vector from an agrobacterium strain and insert it into agrobacterium and actually have the agrobacterium itself transform the tree for you with the traits that you would like. So how we do that is we develop a agrobacterium vector. So right here we have what we call a promoter, and this is a genetic switch. It basically tells the plant how much, where to make, when to make this enzyme that follows. So here's the OXO gene. So basically we put that into agrobacterium and then transform the somatic embryos that we collect. So they only can infect one cell at a time. So those somatic embryos that you saw before are collections of thousands and millions of cells. So when we do transformations, we actually are only infecting individuals. So this is a demonstration of the process with a green fluorescent protein. We just needed to initially in the project figure out, again, can we make it to that stage? So that glowing cell right here is an individual event, and we would grow an American chestnut tree from that one original cell. This would be a second event. So there's a huge process of isolating and then selecting only for the transformed individual cell lines. So there are many, many individuals. So why do we only have one that we are currently moving forward with in the regulatory and restoration process? Each transformation event is random when it comes to location and quantity. So one cell could be transformed with the gene of interest, but it could be inserted 50 times. It could be inserted 1 time. It could be inserted 10. It could be inserted in a location on the genome that is not highly expressed. So it could be just dormant genes that aren't very much used by the organism that you're trying to transform. And so therefore you wouldn't get high enough expression of the gene that you're hoping will confer resistance. And so we need to try a different location in the genome. So one event could have the OXO gene up here on this chromosome pair, but a second event could have the location somewhere down on this one. And this area might be better for expressing because it's more accessible than this area over here. And then sometimes, like I said, there are multiple copies. And like I said, the position effect greatly influences how a gene is expressed. Oxalate oxidase expression in chestnuts also varies based on the expression. So here you can see this is some of my work from my master's. Right here is the original line of American chestnut. We call it a Darling 58. And then these are the subsequent generations. So we crossed our Darling 58 with regular American chestnuts, and then we got these 6 nuts, and these have differing expression. And then we cross out again, and there's differing expression as well. Our Darling 58 transgenic American chestnut, we sequenced the entire genome because we also needed to know, are we interrupting any genes that are being expressed for natural purposes in the American chestnut? And what we found was we inserted it into a region that isn't expressed, and the closest gene is 5,500 base pairs away from our insertion site. So we just want to make sure that we aren't affecting natural expression of genes that are already in the American chestnut, and that location won't change based on our crosses. So with no endogenous gene interruption, we were ready to move forward into the actual regulatory approval process. But I can talk about this all day, but what's really important is, did this work? So we needed to test, is it actually resistant to the chestnut blight? And so what we do are called small stem assays. And we basically grow the fungus on a Petri plate, like you saw in a few images previously. And we make a wound in the stem of the American chestnut, and we put a bunch of fungus on there, way more than you would ever get in the natural environment. But we want to have a robust testing system. We don't want to say that these are almost resistant. If there's not too much fungus or conditions are just right, we want to make sure that these things can handle it. So we have wild type Americans. So these are non-transformed. We have our transgenic American chestnuts, and then we have Chinese chestnuts, which are the resistant controls. So after 6 weeks of inoculation, you can see every single one of the American chestnuts has died at the canker site. You can see some of them are resprouting from the bottom. Just like I was talking about, this is how the chestnut species has persisted today. Our Chinese chestnut, which are the resistant controls, which ideally we would want our Darling 58s and Darling 54s to be performing at least as well as a Chinese chestnut. You can see that a few of them still died. So even though these are resistant, because we put so much fungus on there and they're so small, it really puts a hurting on these trees. And here you can see our Darling 54 American chestnut. They were all healthy. They didn't wilt post 1 year, and we ended up planting them out in the field, and they're still there today. So now that we have transgenic American chestnuts that are resistant, we got to produce a lot of them. So one of the ways that we do that is through tissue culture. And so we can produce clonally these trees in a gelatin media with plant hormones, sucrose, and everything a plant needs to survive, and produce these trees, get them into rooting, and then put them into highlight growth chamber production. And basically what we discovered here is that an American chestnut does not mature sexually until 7 years, where it starts producing male flowers, and then around 7 and 9 years before it starts producing female flowers. we were trying to accelerate growth, and we started putting chestnuts into highlight growth chambers to try to get them to grow faster for planting outside to give them better survivability. And it turns out we can actually induce flowering of male catkins and female flowers in under a year. So we're able to actually produce transgenic pollen every year from the crosses that we made the previous year, instead of waiting 7 years, 7 years, 7 years. So this was a huge breakthrough for us, and it's actually been able to allow us to produce way more chestnuts than we were initially planning when this project was conceived, and actually allowing us to scale up in our lifetime to actually restore the chestnut to its historical range. So here's some pictures of chestnut catkins in the highlight growth chamber. You'll notice that the trees look pretty poor, and it's because we're essentially torturing them, which is sad to say, but they are our martyrs. We put them under 16-hour photo periods, and we never let them senesce. And the combination of the highlight treatment and the stress of never going into senescence makes them produce pollen faster. So then we collect that pollen on glass slides and store it in our -80 freezer after desiccating it, and it remains viable for the next year's pollination season. So we don't need to time our pollen production exactly to when American chestnuts are flowering out in the field. We can actually produce pollen year round and store it and maximize our crossing possibilities. So then the pollen you can see on the glass slide here, we just rub that gently onto the stigmas of the chestnut burrs and then rebag them, and next year we'll have, or later that year we'll have crosses. So this is what American chestnut pollen looks like under the SEM, which is a scanning electron microscope. I just include it because it's cool. And now we are producing so many chestnuts that we actually have to test which ones are transgenic and which ones aren't. Just like with people, trees inherit half their genes from their mom, half their genes from their dad. And since we're pollinating with transgenic pollen, only 50% hypothetically would be containing the OXO gene to confer resistance. So we do that through a histochemical assay where we use these hypodermic needles, but we've actually switched over because it's cheaper to Claire's ear piercing needles, if anyone's interested. And we pierce the endosperm of the nut tissue. So the embryo's up here, we don't want to damage that. We can collect the tissue from the endosperm here on the sides, and we place it into this solution. If it produces hydrogen peroxide, it turns this black color, and that's how we know it has OXO. If it stays white like this, it's a negative tree, and so that's just a wild type American. So now that we're starting to do these outcrosses, we have T1, T2, T3 generations. So each subsequent pollination years, we need to continue testing for resistance. We need to make sure that these guys are actually going to maintain and keep their resistance throughout subsequent breeding generations, because if they're not, it's not a viable form of strategy for restoration. So here you can see 2 transgenic trees. These are T1 nuts. So these aren't produced from tissue culture. These are just nuts that we cross-pollinated and stored in the refrigerator and then planted the next year. You can see the cankers are there. They're very small. There's orange tinting on the outside of these cankers, but they're not girdling the tree. They're not sunken in like their non-transgenic controls, and they're even performing better than the Chinese resistant in this example here. We have continued to do more of those this year. So these are some of the results from this year's stem inoculation. So here you can see the wound that we cut into the stem. We put the fungus that's on the agar onto that wound site. And this is 8 weeks after inoculation. Again, you can see that canker, but it's not expanding. It's kind of contained. The fungus is still living on there, but it's not girdling the tree and killing off the tree. And then this is a full sibling. So this is from the same cross, the same parents, but this one contains the OXO and this one didn't. So this one just got unlucky and didn't get the OXO gene from its parents. And as you can see, the canker is drastically different, and it will girdle the tree, and that branch will die off easily by next year. Here are some more, just to show you some of our crosses. Again, cankers are small on the transgenics, not on the non-transgenics. So on this one, we like to show it because we inoculated this tree. This is the non-transgenic sibling to this one. We inoculated this tree earlier this year, and it completely girdled off and killed that branch even before the growing season was over. So we're doing a very harsh test. This wouldn't happen in a natural environment because we wouldn't be it wouldn't get that much amount of Cryphonectria in just natural infection. And to demonstrate that we're actually not doing anything to the fungus, it's important to prove that we can re-isolate this fungus from these cankers that are on transgenic trees. So we cut off a piece of this canker from a transgenic tree, and we put it onto the agar media to make sure that we can actually grow the fungus again. Because if we're affecting the fungus, we wouldn't want to go forward with restoration if that were the case, because we don't want to have any fungicidal aspects to this. So we're able to re-isolate just like we can from the Chinese chestnut. And yeah, just one more example of that. So now we have demonstrated all of this. We began our regulatory process going through the USDA, FDA, and EPA to allow these trees to be planted in the wild. Right now, we can only plant them in permitted locations where everything's fenced, everything's controlled. We don't let these trees flower naturally. We wouldn't want it to escape preemptively. We also are going through the FDA for food and feed because people are going to eat these. Is it safe? Does it change anything about the nutritional capacity of these nuts? And then the EPA, we are going through the process with them because they manage any environmental protection agency. We're going to be changing the environment. So we just need to go through the environmental impact statement with them and go through any reviews that they might have. We're estimating a 1 to 2 years before we're able to distribute, assuming everything goes well with these agencies, which it has been to this point. And because it is going, the reason it's going well is because we have a ton of collaborators that we are doing a ton of non-target comparison studies. I'm not going to go through all of these because it would take too long, but the one I do want to highlight that everyone asks about is how are we going to be affecting other funguses that are in the environment? So mycorrhizae. They provide symbiotic relationships with natural tree species in the forest ecosystem. It is a great relationship that provides both benefits for the tree and the fungus. And the biggest thing we wanted to make sure is that we're not eliminating or killing off anything regarding those. In all of these studies, bee feeding, tadpole feeding, aquatic insects, germination rates, mycorrhizae, there are no enhanced risks or differences between hybrids, American chestnuts, Chinese chestnuts, and transgenics. They're all statistically the same. We've also started recently long-term ecological research because this isn't a thing that's just going to be, oh, we got it and we'll plant the trees and it's over. We're going to be reverting Eastern US forests back to what they were 100 years ago. How is that going to change what has evolved currently? So we have started 3 different ecological demonstration forests, what we call them. One here in Syracuse, New York. We have one down in Penn State. We have one in Virginia. We also have a 4th one that we just set up in Maine. So this is going to be a high value research for decades as we study American chestnuts in these demonstration plots, as well as the transgenics. Some of the next steps that we're working on, we are trying to refine our transgenics in that our resistant transgenics right now that we're developing, our OXO gene is always on and it's expressed everywhere because we want to maximize resistance. But is there a way that we can tone down some of that expression or make it more targeted that will still confer resistance, but doesn't have such a cost and metabolically or energy-wise for the tree? So we're working with poplar promoters, so the gene switches that are only inducing expression when the tree is wounded or in the presence of a pathogen. So we have some of those that we're producing crosses with right now. We're also working with CRISPR to enhance gene expression in American chestnuts that might confer resistance to the blight. So there might be a gene in American chestnut that's already there that just isn't expressed or it isn't on. And if we were to turn that gene on and express it at a high enough level, can we develop blight resistance? We're also working with all of our partners across the Eastern US for increasing diversity and maintaining germplasms of all the different varieties of American chestnuts. So as you can imagine, an American chestnut that evolved in Georgia is going to be different genetically than one that evolved in Maine or Canada, and so on. Something that prefers to grow on top of a mountain in the Appalachian range might be different than something that wants to grow in the lowlands. So we're trying to maintain and capture all the existing diversity that is still out there from the trees that are surviving from their root sprouts. There's also another chestnut pathogen called Phytophthora. It is a soil-borne disease. This affects American chestnuts, but it's a bigger problem for Europeans and Chinese where it's more prevalent. Luckily, up north right now, our winters are still cold enough that it kills off the Phytophthora, and it hasn't moved up with the temperatures quite yet. But we also have Phytophthora down south. So a big thing that the TACF has been working on is making crosses with B3, F3s for Phytophthora resistance, because that seems to have resistance based on just a couple of genes. So we're doing a ton of crosses of our transgenics with the Backcross breeding program to try to enhance resistance in both the chestnut blight as well as Phytophthora. And we're also identifying all of these resistance genes that are in the Chinese chestnut and the Japanese chestnut and isolating those and putting those into transformation vectors and transforming the American chestnut with Chinese chestnut genes to try to enhance resistance that way as well. So here's our shameless plug. How can you help? You can join the National American Chestnut Foundation. They have newsletters, they have plantings, they have tons of activities and outreach that are great for anyone who wants to get more involved. And we also have a local New York chapter branch. We just had our chapter meeting last Sunday or Saturday, and 50 people came out, all enthusiasts that have their own chestnut orchards that have supported us from day one when we started this project. And they're a great group of people that can help you figure out how to grow chestnuts on your land specifically. They provide nuts. They're a great group of people. You can follow us on any of our social media stuff. You can find all of that at our esf.edu/chestnut webpage. You can plant mother trees. Once this gets once we get regulatory status where we're allowed to distribute pollen and distribute trees, if you already have mother trees, then you're going to be first in line to get some transgenics because you'll be able to cross your trees right away and begin to actually produce blight-resistant American chestnuts. And we also have all of our petitions and documents on our USDA, EPA, and FDA websites. So those are all publicly available that you can download, read, do public comments on, and show your support that way as well. And with that, I will leave it there and take any questions you might have. Thank you.
All right, so if you guys have any questions, just feel free to type them in the chat and I will read them out and I'll put them to Dakota. So the first question is from Tina Chi, who asks, how do you tell if a chestnut tree is male or female?
So yeah, I probably should have explained that. American chestnuts are dioecious, so they produce both male and female flowers. So, you would think to yourself, then why can't my tree just pollinate itself? They're self-incompatible. So you have to have two chestnut trees, just like you would have to have like two apple trees or something like that. And but the American chestnut produces both male and female, and they're usually both on those catkins. So the female flowers will be at the base and the male flowers will be at the top.
Okay, next question is from Simone Montgomery, and she says, a friend of mine was able to sprout some chestnuts he acquired from a gentleman from the American Chestnut Foundation in 1984. And he raised them and I guess planted them in a secret location in Letchworth State Park. Do you know if anyone has ever found this grove and if it survives to this day?
So we have a ton of collaborators, and one of the biggest things that we've been trying to do is really engage citizen scientists. And a big thing that we're doing with that, there's a couple of apps called iNaturalist and also TreeSnap. And people can go out into the woods. If you spot an American chestnut, you spot some birds, you find that tree, you can actually upload that location. There are so many locations that I cannot specifically say. I wouldn't be surprised if someone has found those trees, but I also wouldn't be surprised if they might have found just different American chestnuts that are persisting on their own from stump sprouts. But yes, we get a ton of help from everybody finding new wild types to incorporate into our breeding program.
All right, so everyone who's here tonight, you know your mission. Get iNaturalist, go to Letchworth State Park and find that grove, and then let us know so we can credit you with the discovery. Okay, next question comes from Patty Black, who's actually the president of the Strathmore Speakers, and she asks, is anyone working on applying these techniques to other endangered trees like the elms and trying to slow Dutch elms disease, things like that?
So it's funny that you mentioned elms. We actually have a satellite project at ESF, and we have a very generous donor from down in New York City that just loves American elms, and he's actually been funding us to develop a Dutch elm disease and elm yellows resistant American elm tree. We're still in the early stages of that when it comes to production and validating genes and stuff like that. But the big issue there is for us that Dutch elm disease, or it's elm yellows, one of the two, is transported by an insect. So it's very hard to do controlled inoculation. So the only way you can really figure that out is by planting a possibly resistant American elm in an area that already has elm yellows and Dutch elm disease. But yes, we are working on that. We have transgenic elms that we're moving through the pipeline, and we are still identifying genes, trying to optimize that. There are people who are working on emerald ash borer. I think it's Purdue. I think that those are the only two examples I can come up with at the moment, but a big limiting factor in this is a lot of people can come up with ideas and go through this process, but no one wants to pay for it if you can't release them, if you can't use them. And so we're really hoping that our project can be kind of like the front runner when it comes to setting the mold or setting like a stepwise protocol to get a tree through that. So we definitely expect a lot more research to be done throughout the US if we're able to achieve regulatory status.
Okay, next question. Well, I guess it's more of a comment. It's from Elizabeth Costello, and she says, it's incredibly interesting. You must look forward to going to work every day because it's such an exciting project. What's a day in the life of the lab manager like?
It's great. It was a little weird during COVID, as you would expect, because I was basically the only one there. And, we teach plant biotech class at ESF. And what's really cool about that is all the students get to do their own individual projects, and their stuff is, because we fund it directly, their stuff is directly related to chestnut research. So I really do enjoy teaching undergraduates and having their projects come to fruition, because if they do, they help us, because we might use that research in the future. So we do have quite a few transgenic lines that were generated by undergraduates or master's students that they have their little initials next to the strain name. And so I very much enjoy that. I love being able to work both out in the field and in the lab. That's a rare thing for someone who's in science. You're usually pretty set on one or the other. So I love the variety. Yeah, it's a great job.
Awesome. This question is from me. Are there any epigenetic effects from the accelerated pollen generation regime? Those tortured plants that you showed us.
So yeah, that is something that we can't determine quite yet. But because we are outcrossing with so many diverse families, and it is pretty consistent that almost every tree in those conditions will do it. It's not like only certain varieties will produce pollen underneath those lights. So we're not selecting for early flowers or selecting only certain varieties that would be optimal for those conditions. But that's something we can't really know until we have the full genome sequence, and then we have the resources to actually start sequencing all of the genomes of all of our crosses throughout. But any of those effects that might pop up are going to be diluted out very quickly, and are being diluted out right now as we outcross to all of these diverse family groups. So yes, that is a concern, and we've thought about it, but we don't expect it to be as predominant as maybe you might think. And then also, it's not like we're also selecting for a trait that's not advantageous. Earlier flowers are just fine when it comes to trees. So yeah.
That's awesome. And then once, if anybody else has any other questions, go ahead and type them up in the chat. If not, I have one further question. So once you start distributing, where are you going to be distributing? Like, how do you decide? Like, well, if it's assuming everything's approved, how are you going to go about getting the trees out into the wild?
So, like I said earlier, citizen scientists are a huge part of that. So we have a database that we maintain at ESF of everyone that has flowering American chestnut orchards in basically any relevant chestnut state, as well as New York State, obviously. And we basically keep track of that. It's basically a waiting list. And if you have a flowering American chestnut, we will send you pollen when we're able to, when we have enough. And what we'll do is we send you those pollen slides and you do your own controlled pollinations. We also have been doing pollination workshops, which is something that you can sign up each year. Basically what we send you, if you have a flowering tree, is a pollination kit where you can practice doing your own controlled pollinations with just regular American chestnut pollen. And if you demonstrate success with that, you will be the first one to get as much pollen as we can give you. We're also setting up educational plots and production plots all across the eastern US for eventual distribution, where we'll distribute nuts from our crosses for plantings for production and also restoration sites. So one of the big things that we're working on is identifying already disturbed habitats, because we don't want to selective harvest. We don't want to go in and change anything drastically right away. So one of the big things, because American chestnuts love well-drained soils, is planting in old mining mountaintops in the Appalachians for coal mining, things like that. So we're working on setting up partnerships along those lines. And also just everyone who supported us in the past, TACF members, New York chapter members, they're all on lists as well. And you get on those lists when you join and send an email, and we will distribute. We're trying to balance the restoration with also the public interest and also the support that we get. So we're also distributing just to people who want to plant them in their backyard and have them for whatever purposes that they might want.
Cool. Awesome. So that might answer the next question, which is from John Turpinning, who said that you can attract wild turkeys to your property with chestnuts. How would he source some chestnut trees? So
Oh, well, yeah, the American
up,
Chestnut
so-
Foundation.
Yep.
I
Sign
would
up.
definitely reach out.
Cool. Awesome. And then I guess last comment is also from Patty Black. I guess there are some chestnut trees in Onondaga Park, but of species unknown. So
Hmm.
interesting.
Yeah, I'm not sure if we planted those or what. I bet, like, I would imagine they're either Europeans or Chinese or hybrids, one of the two.
Cool. Awesome. All right. Well, if no one has any further questions, I want to say thank you to Dakota. That was a fascinating and fantastic talk. Usually when we discuss humanity's ability to control nature, we're talking about something really negative, but this is a really, really positive application of that knowledge and that technology. So thank you for all your hard work and just, yeah, amazing and fascinating project and fantastic presentation. So thanks for being here tonight.
Thank you so much. I really appreciate it. It's been great talking to you.
Awesome. And for anybody who's interested, when we post this video on Onondaga Free's website and on the Strathmore Speakers website, we'll have links to a lot of this, the organizations that Dakota mentioned too. So if you can't find it on, the general interwebs, you can always, go to our site and we'll have links there. All righty. Well, I am going to turn it back over to Mary Beth, who's going to take us out and tell you all about our speaker for next month. So take it away, Mary Beth.
Many thanks to Dakota Matthews for his presentation tonight. We hope you can join us again on Thursday, November 10th for an evening with historian Dennis Connors. He will recount the tale of the Onondaga Arsenal and how our community treasured, remembered, and ultimately lost a unique link to its past. Updates on the series can be found at our website, strathmorespeakers.com. Thank you all for joining us tonight, and we hope to see you again next month. Bye-bye.